again, for those of you who are joining on, we're continuing from our earlier session where we talked about some of the basics of IPA. Now, in this later section, we're now going to talk about, for those of you who have worked with IPA, perhaps entered your data into IPA, generated some results, and are perhaps unsure how to parse through that information, we're going to focus on that this afternoon. I will mention, again, just like the morning session, we will be recording this. We'll be able to distribute it afterwards, so you'll have something as a reference. And I'll also make a point to just kind of flag up, again, areas that would be of relevance as you're kind of going through a data set for the first time and trying to make sense of, what is most relevant for your given study. So with that being said, let's go ahead and begin the second session. Again, it looks like most of you have already been on. I'll just give a quick reminder once again that if you have any questions as we go through this, please do go ahead and submit them through the Q&A button. I have my colleague Sean Prince on the line. He will be available to help answer questions. And I will try to leave at least some time at the end to perhaps tackle questions that existing users have. Now, that being said, if you have very simple one-off questions, perhaps the chat is the most appropriate that you can do there, or if you have kind of bigger scale questions, we can parse that through that at the end of the presentation, this section. So for this afternoon session, what I just wanted to do is begin discussing the core analysis. And really, this is kind of what gets at most of the key biological endpoints when you bring in a data set into IPA. In particular, we'll walk through some of the key components of that core analysis. We'll discuss how we can identify key pathways that are being modulated in our data set. We can look at predicting regulatory molecules. And what we mean by this is we observe in a data set typically a set of expression changes. Now, what potential molecules could there be that are driving those observed expression changes? Conversely, we'll then look downstream. We see sets of genes changing in our data set. Again, now the question becomes, what is the impact of that expression change in our study? We'll also touch on a couple tools that tie into kind of how you can use your data set to build some downstream hypotheses for further experimentation. Lastly, we'll focus on comparing data sets internally. We will, in Monday's session, talking about how you can compare your data to public data sets that have been pre-processed in IPA. But for now, we're talking about internal comparisons. This will be in the context of things like, again, as I mentioned earlier, different treatments, different time points, different disease states, or even things like different cell types within single cell data. So we'll look at a couple examples that reflect those different types of studies that you may have. Lastly, again, we'll summarize. We will, again, try to leave a little more time if I can go quick enough to get through for a little bit of live Q&A, and then again, we'll share the resources at the end. So with that, I wanted to go ahead and begin. And from here, just kind of taking off from where we ended off last time. Again, the key thing for those of you who have worked with IPA, you know that the data that we take in as kind of our starting point for analyzing a data set is not, let's say, a matrix of gene expression or FASTQ files from RNA-seq data, but rather the results typically of a statistical analysis, where we have a study using RNA-seq, using proteomics, using metabolite data, or using even phosphoproteomics, where we're doing some kind of comparison of an experimental group relative to a control group. And as such, again, we'll get standard metrics beyond kind of our molecules of interest from this analysis. We'll get gene symbols, but then we'll get fold changes, p-values, right, letting us know what genes are significantly changing and in what direction are they changing in our experimental group. This information is brought into IPA. We looked at the upload earlier. We would take this, we would run this in the core analysis, and the key biological endpoints that we're going to get from the singular analysis are, first and foremost, the namesake of the software. So what pathways are changing? Taking any given pathway that's in our knowledge base, do we see an overabundance of differentially expressed molecules in our data set within that pathway? That's going to tell us what is biologically important. Here, that's relayed by the length of the bars themselves. And then subsequently, we're going to take this pattern of expression that we have in our data set from a fold change or a log ratio, and we're going to utilize that to compare to our directional information from the literature to actually try to answer the question now, how overall is that pathway changing? And we saw earlier today, these colors of orange and blue, these are going to relate back to predictions of activation or inhibition. So we're going to make a statement overall how the pathway is changing. But beyond this then, we have the ability to interrogate pathways at a much finer level of detail. And in fact, we can choose any pathway that we identify from the analysis. We can open up that as a network. We can understand, here the molecules outlined in purple are where in the pathway I see expression changes in my data set. Again, it could be at the gene level, the protein level, what have you. What we're going to do then is overlay green and blue colors here, or sorry, green and red colors. So the greens are observed decreases. I think there are a couple of reds here. Those are observed increases in our data set. And we can actually use that expression to make a prediction on a secondary level, which is we have perhaps assessed overall if a pathway is activated or inhibited. But based on the molecules we see changing in the pathway, can we predict how other molecules that we don't see changing through our assay, whether that's at the level of transcription or protein levels or metabolite levels, how those other molecules might be changing? And so you'll see on this network other nodes with orange and blue molecules, and these would represent the predicted individual molecular changes in the pathway itself. So we can overall get an assessment of what pathways are important, overall how they're changing, and then how our observed gene changes or protein changes in that pathway can influence other molecules in that particular pathway. Another endpoint that we're going to generate is, again, as I mentioned in the introduction here, a scenario where we have this observed set of expression changes going on, let's say in our experimental group. And we want to ask the question, what might be drivers of those expression changes in our study? So given the fact that in our knowledge base, we know for any given regulatory molecule, what are its downstream targets, we can ask the question for each regulatory molecule, do we see an overabundance of targets in our data set? If we do, that's going to lead us to suggest that that regulator may be involved in controlling our observed transcriptional change or our observed protein change. Then, just like with pathways, we're going to ask a follow-up question. If we deem a regulator important in our expression programming in our system, then we're going to look at the pattern of expression in our data set. Again, utilize that relative to a pattern we derive from the literature to then try to predict, are those regulatory molecules activated or inhibited? And in this context, again, when we think about these regulatory molecules, obviously these would be targets for therapeutic intervention, perhaps. These would be biomarkers, right? These could be a number of things, but these are the key molecules kind of facilitating the change in our system. Now, we ask a question converse to that as well. We say, when we see a set of molecules changing in our data set, can we associate these molecules with a given phenotype or function? So what is the consequence of our expression change? And in short, this heat map that we're visualizing here, each square on this heat map is going to represent a particular term. It could be a function like apoptosis, necrosis, or it could be an indication like glioblastoma, things that we looked at earlier. The size of the box is going to represent the significance of enrichment of data set molecules. The coloring, again, is that follow-up question. When we identify what could be the consequences, how are the consequences manifesting? Are they manifesting as an increase or a decrease? Again, this is going to be driven by the observed pattern of expression in our data set. And then lastly for today, we'll touch on other tools that we have in place that we can use to generate hypotheses. So taking the last two components of the core analysis where we've identified perhaps regulatory changes and then functional consequences, we can use algorithms to put those together into a cohesive mechanistic network where we establish perhaps a key set of regulatory molecules at the top row in this network that will work through a subset of our changing genes or our changing proteins in our data set. And those genes or proteins in turn will then influence a key set of functions downstream in our system. So these are all some of the endpoints that we can generate through the core analysis and IPA. We're going to walk through each of these in a little bit of detail.
QIAGEN.
Sample to insight.